Well, hi everyone, and thanks for inviting me here today to speak to you about the role of scientific research in marine environmental management. And of course, this is a very broad research topic. And so today I'm going to focus on my area of research, which is on threatened and migratory species. And I specifically work on some of the very large animals, and they're often referred to as marine megafauna. So things like turtles and sharks and whales. So around the world and in Australia, many of these species are under threat. For example, from pollution, including plastic pollution, chemical pollution from oil spills, noise and light pollution are big problems. Global fisheries are a big threat. So not just being caught as bycatch, as some of the turtles and cetaceans can be, but also being the targets of fisheries themselves, like a lot of the shark species. And then ship strike can be a big issue for some of the very large ones that spend time at the surface, like the large whales and also whale sharks. So besides being threatened by many human activities, all of these animals have another thing in common, and this thing that they have in common can make them very hard to study. And do any of you have any idea about what that might be? No? Well, I can tell you what it is, and it's pretty obvious when I say it. It's the fact that they all live in the ocean. So they spend most of their lives underwater and far from shore where we can't see them. So, for example, animals on the land, we can see what they do, but for marine animals, especially some of these ones that I'm talking about, because a lot of them are very migratory as well, so they travel hundreds or thousands of kilometres sometimes, and as I say, far from shore and under the water where we can't see them. And so that's where my field of study comes in. And my field of study particularly is as an animal movement ecologist. So I study where these animals go and what they do. And the way that I do this is using these little devices that I'm holding in my hand right now. This is called a satellite transmitter. And you can see that it's got some aerials sticking up. I think you've all heard of a GPS. Well, this little tag has a GPS inside it, and these little aerials on the back here are the receivers for the GPS that's inside here. It receives the GPS signals using these aerials, and then it transmits them through this big one to the overhead satellites, and then I can download the data later, after I've attached this to an animal, of course. And so as the animal's swimming around in the ocean, every time it comes to the surface, when it's got this thing on, it'll transmit its location to the satellites, and that's how I can follow them. And so, as I say, this is one of the main tools of my trade. So I'm just going to play you a little video now that shows putting one of these tags onto a turtle so you can get an idea of what I'm talking about. And so hopefully I can seamlessly share my screen with you. And I'm not going to play the sound on the video, but don't worry, you don't need to hear it. It's really just to see the images. Can you see it now? Okay, so here's a turtle that's coming up the beach, and this turtle here is digging a hole where it's going to lay its eggs. And this is the time that we can access the turtles. Most of the time they're at sea, as I said, but they come ashore to lay their eggs. And so at this time, that's when we can access them to attach the tags to them. So we usually go on the beach at night. Here we are preparing a turtle to have a tag put on it. Here's a little blob of glue that we're putting on the turtle's shell and the tag. And while we're doing this, we just enclose the turtle in a sort of a wooden corral just to keep it quiet while we do this. And then once the glue has set, then we release the turtle, and it can then go back to sea and go about its business with this satellite transmitter on its back now, as you can see. So eventually that transmitter falls off, and the turtle doesn't have any impact from the transmitter anymore. And so now I'm going to play you a little animation of what those turtle tracks look like from all the turtles that we've tagged in Western Australia over the years. And so here it is now. Hopefully you can see that. All these little squiggly lines are animations of where all these turtles have gone. And you can see that some of them go really far and wide. That one there going up to Indonesia, to the island of Sumba, and others going to the Northern Territory. I'll play it again because it was quite quick. These are actually green turtles. The one I showed you in the video was a hawksbill turtle. So you can see one of them going to, all the way to Cape York there in Queensland, to the Northern Territory, to Indonesia. So they really do travel far and wide. And so knowing where these animals go has really important implications for how we manage the many threats that they face in the ocean. And I'm now going to try and share another screen with you, if I can manage to do that. Hang on a minute. And so hopefully I'm now sharing with you a PowerPoint screen, and if I can get the thumbs up, yeah, okay. So in case any of you were watching that and thinking my job is really just all about fun and going out onto beaches and attaching satellite tags to animals, there's also a serious side to my work, and of course it is fun, those things I've just showed you, but with all the data that comes back from these satellite tags, we need to analyse them and turn them into outputs that the wildlife managers and the policy makers can use to make their decisions about how to manage these animals. So what I'm showing you on the screen now is a map, and the orange and yellow polygons, sorry, I'll just make it bigger. The orange and yellow polygons that you can see there are the areas that we've calculated the turtles spend time during the nesting season. So I showed you before in the animations that they travel very far and wide after they finish nesting and they go to their foraging grounds. But prior to that, they spend a few months in the vicinity of the nesting beaches. And I'm showing you some of these areas now. So Barrow Island, the Montebello Islands, and these orange and yellow polygons are the areas that I've calculated from that tracking data where they spend the most time. And now these dotted lines that you can see around these areas are areas that the Australian government has designated as areas to protect the internesting turtles. And they've designated a 20-kilometre spatial buffer around the nesting islands. And so what our data has allowed them to do is to see whether that buffer that they designated to protect the turtles is adequate. And you can see that it is, because it encompasses a greater area than what the turtles actually use during the nesting season that we've calculated here. So this is just one of the ways that this data can help us to manage the threats that these turtles face. And look, now I want to switch gears a little bit and talk about another area of my research. And I want to talk about this particular piece of research, which was looking at the impacts of light pollution on turtle hatchlings, because it's got a really measurable impact. And it really displays quite nicely the role of scientific research and environmental management, which is what I'm here to talk to you about today. So turtle hatchlings, when they hatch out of their nests on the nesting beaches, this is a turtle nesting beach here at dusk, And we know it's a nesting beach because these are the tracks of the female turtles, the tracks that they make as they come up to make their nests. And when the hatchlings emerge, they need to very quickly find their way to the sea. And once they get to the sea, they need to very quickly swim offshore because there are a lot of predators on the beach and in the near shore that want to eat them. So unless they do this quickly, they will fall prey to those predators. And the way that they find the sea after they emerge is using light cues, natural light cues. And they do this by moving away from the darker horizons, which are typical of landward directions, such as dunes. And they move towards the lower, brighter horizon above the ocean. And so light, because natural light is so important to turtle hatchling sea finding, artificial light sources can interfere with their ability to find the sea. So hatchlings can be drawn towards lights and they might not find the sea, or sea finding might be delayed. So they might be just confused and walk around in circles. And of course, as you all know, there is a lot of light sources on the coast. These are the areas where people like to live, like us here in Perth, of course. There are street lights, there are houses that have their lights on at night. In some places, there are many tall buildings with a lot of lights. So this is a problem. So what are the consequences of failure or delay for these turtles to find the sea? Well, as I said, predation is a problem. It's a natural part of life, of course. Sea turtles don't really have, well, the hatchlings don't have many defences. So naturally they're the prey of crabs and seagulls, as I've shown here. But if they're delayed by lights, causing them to be confused and linger, then this predation could be even higher. So even more of them could be eaten on the shore before they even hit the sea. And of course, once they get to the sea, their problems aren't over because there are a lot of other predators in the water, like this shark here who's got a turtle hatchling in its mouth. So what we're finding now in the modern world is that light pollution is not just restricted to the coastline. What I'm showing you here is a satellite image of artificial light at night time. So this is in blue, it's a map of the Pilbara region. Hopefully you can see that's Karratha there. And so all of these white blobs are light sources that show up on the satellites at night. So of course these ones are on land. This is the town of Karratha. But you can see that many more are at sea. Cape Lambert here, there's a big port for transporting iron ore overseas. And so this is the shipping anchorage off Cape Lambert. Here's another one off the Dampier Archipelago. This is Barrow Island that many of you would have heard of, where there's a large gas plant. Look at the amount of light that is present at night. So this is a very remote area where not many people live, and yet there's a lot of light here. And this is a problem because all of these circles I'm showing you now on the screen are where turtle nesting beaches are, where the little baby turtles are emerging from their nests, having to get to the sea, and then once they get to the sea, having the possibility to be confused by all these lights in their environment. So what's the problem here? So most of what we know about most of what we know about light pollution and the impacts to turtles comes from the short and on land part of their dispersal, like I talked about before, when they emerge from their nest and they transit from their nest to the seashore. But we know very little about what happens to them once they actually reach the water. And that's because we can't track the turtle hatchlings in water like we can the adults, because remember this tag I showed you before, look how big it is. This is bigger than the actual turtle hatchling. So of course I can't put this on a hatchling because it'll just sink to the bottom of the ocean. There are some that are a bit smaller than this one, but they're still about the same size as a turtle hatchling. So they can't actually carry these devices in order for us to know what happens to them once they go to sea. So what we did was we used this other form of tracking called acoustic telemetry, and this is often used on fish. And how it works is we use these tiny little acoustic transmitters, and we attach them to the underside of the little baby hatchlings. They only stay on for about a day, so don't worry, they fall off and then the turtles can go about their business again. But what they do is they emit a high frequency sound that it can't be heard by you or I or the turtles, but what it's heard by are these acoustic receivers that we place in the environment. So we moor them on the sea floor, and if we have a number of these different receivers in the environment, then we can track them. So if the ping from the little tag on the turtle is heard by at least 3 of these receivers, then we can actually calculate a position using triangulation. So this is a really, as I said, it's a method that's been used on fish. So we really wanted to see if we could use it to answer this question of whether light is a problem for turtles, swimming turtles, when they reach the sea for the first time. So now I'm going to show you some tracks, some animated tracks, like I showed you before, of the satellite tracks of the adult turtles from the baby turtles. So we did this at Ningaloo on green turtles, and there's some videos here. On the left-hand side is the tracks of the turtles. I'll play it in a minute. We released the turtles on the beach. So here's the beach here. Here's the vegetation behind the beach. Here's the water, of course. And each of these little white circles is an acoustic receiver that we've placed in the water to hear the pings from the turtle hatchling tags. And so we did the experiment over 2 nights. On this first night, we didn't have any light. It's called ambient. So ambient means in natural conditions. And then on the second night, we did the same thing with some artificial light, which was present on a boat that was moored on the edge of the receiver array. So I'm going to play the videos now and you can see the difference. So in the ambient light treatment, where there's no light, the hatchlings all just fan out. When we had artificial light present, all the turtles were very strongly attached. So from when we released them on the shore, they all swam directly to the light. I'll play it again, and they lingered around this light source. So you can see that we've shown really clearly that when you place artificial light in the turtle hatchlings environment, that they're very strongly attracted there. And they stop doing what they're supposed to do, which is like I'm showing on this video on the left, which is heading out to sea and out to the pelagic zone, which is where they spend the rest of their life as juveniles. So next, we wanted to see how far away we could draw the turtles. So for this experiment, we used a jetty, and this jetty is on Thevenard Island, which is not too far from Barrow Island, that some of you might know. And so we wanted to know how far we could draw them from the light source, but also we wanted to try and see if we could actually measure predation. So we have this theory that if you attract turtle hatchlings with artificial lights in the environment, that stops them from going about their natural dispersal behaviour. And the theory is that that increases them to predation, but we wanted to really measure whether that was actually occurring. And at this jetty, at Thevenard Island, like many jetties around the world, fish congregate under them. And we know this because fishermen go and fish off jetties all of the time. And some of you yourselves may have fished off a jetty. It's a good place to catch fish, right? So fish are some of the predators of turtle hatchlings when they're in the water, of course. And so we use this jetty because, of course, there are lights on jetties and because the fish congregate under there, and we thought we'd be able to measure predation. And so we had 3 receiver arrays this time, and the first one of them was at the jetty, the second one was 200 metres away from the jetty, and the third one was 400 metres away. And again, we did the experiments where we had lights on and we had lights off, and we did this over 3 nights, and we had 10 turtles in each of these conditions. And that's just to show you what the jetty looks like at night when the lights are on. So what we found this time was actually really surprising. Only 30% of the turtles that we tracked returned tracks that we were expecting. And what do I mean by what did we expect? Well, we expect that the turtles transit the shore, the nearshore area very quickly, as I showed you before, because they're supposed to just go out to sea and spend the rest of their development in the pelagic zone. So they don't hang around here. So again, I'm going to show you a video. Each of these white circles on the map are the receivers that listen for the pings from the tag. And this red dot here are the turtles as we're releasing them at the jetty. So you can see that they swim very quickly out of our array. Once they're out of our receiver array, we can't hear them anymore, but they're going out to sea. So they're only in the array for a very short period of time. And this is what we expect to see. But as I say, we only heard that, or saw that for 30%. And so what happened to the other 70%? So I'll show you that now. And the data suggests that most of our turtles were predated. And you'll see now when I play this video that it's really very obvious that these are no longer turtles. What these are now is our turtles with tags have been eaten by predators, and the tracks that you're looking at now are the turtle tags inside the stomach of a fish predator. You can see the video's changed to daytime now, and during the day, these fish that ate our turtles with the tags on them then went and resided under the jetty. So they rest there during the day, and at night, I'll just play it again, they come out and they zoom up and down eating our turtles. You might think this sounds pretty bad, but of course, like I said, predation is a natural part of life for a turtle hatchling. And what we've been able to record with these tags is indeed that natural part of life. But what we're showing is that these jetties, not only do they have lights on them that attract turtles, but the lights also attract the fish predators, and they provide habitat for these fish, and might cause them to be in greater numbers in these areas around turtle hatchling beaches. So what were the predators? Well, we don't know for sure because, we couldn't observe this. It all happened in the dark. And but what we do know is that we put a remote video under the jetty, and we saw lots of these things. They're called mangrove jacks, and they're abundant in schools under the jetty. And they're also a species that the fishermen like to catch. They're a good eating fish. And we actually got some fish frames from the fishermen. And when the fishermen gutted the fish and opened them up, lo and behold, look what's inside the bellies of these fish, our turtle hatchlings. So we've been able to show that these fish are probably the likely candidates of the fish species that ate our tagged hatchlings. So look, what are the conservation implications for this? So these, the jetty where we did our experiment is a very little one. It's only 70 metres long at Thevenard, but around Australia, or especially in Western Australia, there's some really large port developments, and these port developments are used to transport a lot of our resources like oil and gas and iron ore offshore, and some of these are 1,000 metres long or 2 kilometres long. So, these structures are all lit. If our little jetty can harbour lots of predators, imagine what these ones could be doing. So, these jetties have the potential to impact the number of hatchlings that are being recruited into the population. So obviously, if all the babies are getting predated, we won't have any baby turtles becoming adult turtles and coming back to nest. So it's a bit of a problem. So look, you know, I talked about a lot of the fun, of course, of doing the field work and working with turtles, but there's a lot of hard work involved to analyse all this data and get it out there where wildlife managers and policymakers can use it. And the way that we do this, and the principal way that we do this, is by writing up our results in the scientific peer-reviewed literature. And so that's what we've done in this case. This is the first paper that we wrote, which was in the Journal of the Royal Society, and then another one in this other journal called Biological Conservation. So that's how the the managers and policymakers can find our research and see it. But that's not all. We also want to do presentations, of course, like I'm doing for you today. Presentations to the general public are really important, but also presentations at conferences. There are a lot of conferences, scientific conferences around the world and in Australia, of course. And then also to the media. Here I am being interviewed by ABC Radio National about some of the research. And then, of course, not just writing scientific literature in the peer-reviewed literature, because that's a little bit inaccessible to the average person, but also making the information really easily available. So in this case, I wrote this little story, which is on the website of the Ecological Society of Australia. It's all written in plain English that anyone can access, which really clearly outlines the problem and the evidence behind this problem and the implications of it. And so, look, that it doesn't stop there. It doesn't stop there at all. Once you've identified a problem and you've done some research and you've published it and you've presented it widely, we also want to know that our research has had some impact. And in this case, we were really lucky in that our research was picked up and included in these National Light Pollution Guidelines for Wildlife, which is published by the Australian Government, by the Department of Environment and Energy. It includes marine turtles, seabirds and migratory shorebirds. And our important research on the effect of light pollution on the in-water part of turtle hatchling dispersal was included in these guidelines. So it's a really great impact of our research. So, you know, to summarise about the role of scientific research in environmental management, I just wanted to summarise all the points that are involved. Obviously, you go out and you undertake the research and to provide the scientific evidence of the problem. In this case, the impact of light pollution on turtle hatchlings and particularly on their in-water dispersal. We need to communicate the results widely, firstly in the peer-reviewed scientific literature, but we don't just stop there. At national and international conferences, to the general public, speaking to the media, using social media, and using the internet, of course, and writing this information in a way that everyone can understand. And success is really when there's a change to policy or regulation. And the ultimate success, of course, is that policy or regulation change has resulted in recovery of turtle populations in this case. And so, do I know if this has been the case? Well, not yet, and maybe we won't know because we're not monitoring all the different turtle populations around Australia. I showed you a map before of some in Western Australia, but turtles are found all across Northern Australia. So Western Australia, the Northern Territory and Queensland, we can't possibly monitor them all. But what we can know is that given we have these regulations now, and if they're followed, there's a really good chance that there's going to be a positive impact here and that impact will flow on to more turtles. Less turtles being predated, less turtles being trapped on land and not being able to find the sea, and less turtles that will have an issue getting out to sea and becoming adults and returning to nest themselves. So look, guys, I'll stop there. It's not always that good to drone on and on. I'd really like to invite you all now to ask me some questions, or we can just have a conversation about what I've shown you today. And yeah, please ask me anything you like. I'm more than happy to answer it.
How does your turtle research compare with our research globally? And has there been a reduction in turtles due to coastal developments?
So yeah, that's a pretty tricky one, and sort of what I've touched on a bit at the end. So we know that all the turtle species are in trouble. They're all threatened. Some of them are endangered. Some of them are critically endangered. And it's from a range of things, not just coastal development. Coastal development's one of them, but also talked about fisheries interaction and a number of other things that impact them. And in other parts of the world, a big problem for them is the taking of their eggs for food, and that's the illegal take of that. So, you know, there are so many different threats. So we can't pinpoint it all to coastal development, but of course, coastal development is one of those threats, and we need to manage it. So, you know, humans obviously use these same coastal environments, so it's, you know, we can't just stop all coastal development. We need to manage the risks carefully, and that's, you know, before I showed you the map of where the turtles spent during their nesting period. And, you know, we can't just protect the whole ocean. What we really need to do is just understand where the most important areas are and where the places are that the turtles are under most threat, and try and make our impacts in those areas. Does that answer your question?
Yeah. If you could choose any avenue of research to be funded, what would you choose to research?
Wow, what would I choose to research? Well, I like turtles, as you've seen, but I also am really drawn to some of these other animals that are quite difficult to study. I also work on blue whales, and many of you probably know what a blue whale is because it's the biggest animal in the world. And recently we have been trying to put some of these satellite tags on the blue whales, and it's a really difficult thing to do. We were successful, and but there's still so much more that we need to do. So I probably would like to do a bit more on them. They're fascinating animals. They spend most of their time underwater, so they're really hard to work on. And in Australia, they migrate from just off Perth. Off Perth, we have some really big canyons called the Perth Canyon, and they forage there, and they travel up to Indonesia to breed. And so, yeah, being able to understand where they spend their time along this migratory route is what I'd really like to spend a bit more time
on. Going on with that, is there any developments in the technology of recent times that is kind of like, I know it's very difficult with satellite tags, and you need to make sure that they're friendly to the animal, and it's almost impossible to get data when the animals you know, spend most of their time underwater, which is most of the marine, you know, animals. But is there been any developments in the technology that we're using for this tagging process and other research like that?
Yeah, there has been quite a lot. So this little tag that I'm showing you now, it's pretty cool. It's very small. You know, once upon a time, these tags were huge, and so every year they get smaller and smaller. They're not small enough yet for a turtle hatchling, but they're getting really close. So the smallest tag now that we have is 5 grams. So the average green turtle hatchling that I showed you before, they're about 25 grams. So 5 grams, you know, that's a quarter of their body weight. Still too big, but you know, we're getting there. And pretty soon there's going to be a tag that's small enough. You know, as you all know, everything in electronics gets miniaturized, and so it's only really a matter of time before we have the tags that are small enough to use on these tiny animals, and also a range of other animals. Birds are an animal that, you know, a lot of them are quite small, and we need small tags for them. So, and it's not just the size that's coming down, but also a number of other sensors are being added. So I've showed you this tag that's just collecting information on where the animal is, but there's many other tags that provide other pieces of information. So on how deep the animal's diving, what the temperature of the water is where they're diving, other information about the oceanography around them. Also things, a heap of other different sensors such as fluorescence, which can tell us about the, you know, the tiny animals that live in the water column that are the base of the food chain. So when these animals fluoresce, that gets recorded. So you can say something about the prey field of the animals using these kinds of tags. So, you know, heart rate monitors, all those sorts of things are being added to these tags all the time. So it's a really exciting field. And you brought up a really important point too, which is the ethics of the animal, which is also really important. So all the research that we do has to go through an ethics committee, because we don't want to hurt the animals. We don't want to impact them by carrying these pieces of equipment. So they're always designed to stay on for short periods of time. And in the case of turtles, sometimes they can stay on for a long period of time, but invariably they do fall off. And at that point, you know, nothing remains on the animal to hurt them in any way. And as you can see, they're often quite small. So, you know, the impact of carrying a little thing on a large animal is also small. So it's a little bit like you guys walking around with your backpack. As long as it's not full of books, you know, it's not a real heavy thing for you to carry around, and it doesn't have a big impact on your movement. But that's always what we need to be checking, that the size of these tags is only around 3% of the body weight of the animal, so that we don't impact them too much.
What are the challenges and implications of managing the species that migrates across international borders?
That's a really excellent question, because you saw in the videos that a lot of these turtles did go to other countries. So some went to Indonesia, some went to Papua New Guinea, and some, you know, went obviously to Queensland, which is still part of our jurisdiction. So when they go into these other areas, of course, those other areas have different management regimes to us. And like I said, in some of these places, they take the turtle eggs in an unregulated way. So they take more than the population can sustain. And in some of those places, they also take the adult turtles. So they can take them for food, and they can also take them for the tortoise shell trade, and that's a problem for the hawksbill turtles. So the turtle shell trade, you might have heard of. You know, the shells are used to make jewelry, and you know, in the old days to make combs and sunglasses and all sorts of things. Decorative items, you know, it's an illegal practice in most of the world, but it goes on illegally and unregulated in some of these countries. So it is a big problem. Well, it can be. What we really need to know is what proportion of our population go to these places and spend time there. And so that's why these sorts of studies are really important, because once we know what proportion of our turtle population is going to these places where they might be under more threat, then we can engage with those other countries, the wildlife management agencies in those countries, to try and make some changes. But yeah, it's difficult.
Has there been a rise in global collaborations in research in recent times?
Yes, there has, and that's also very important here because the satellite tags that I've shown you, they're really expensive. This little one here, this one is about $5,000. So you can imagine that if you want to know a lot about a population, you have to put tags on a representative proportion. So just like if I went and randomly picked one of you and measured your height and say the person that I picked out of the crowd was 160 centimetres, and I wanted to know the average height of students of your age, I'd have to measure quite a few, right? Because if I only measured 3 of you and you all happen to be small, then that wouldn't give a good indication of the average. So you know that we have to measure a lot of students to know what the average height of students are. So it's the same here. We have to tag a lot of animals to know on average where they go and how long their migratory paths are. And because these tags are so very expensive and the logistics to go and deploy them is also very expensive, we need to collaborate widely to get the numbers of tagged animals up in order for it to be representative of what the entire population does. So yeah, so we collaborate with people all around Australia and overseas to do that. The animation of the green turtle tracks I showed you, that's from exactly one of these collaborations. So in Western Australia, we collaborated with a number of different research institutions where we all pulled our tracks from green turtles and we analysed them all together. So there were 100 tracks in the end, and using that data we were able to work out the areas that were most important for green turtles in Western Australia. And that's being done more broadly now because green turtles aren't just found in Australia, they're found in Southeast Asia as well and in other parts of the tropics. So it's a really great thing that we're doing now, collaborating globally to understand the entire distribution and the migratory pathways of these animals.
Have you been able to estimate the impact of light pollution on hatching mortality rates at a species level?
Yeah, so we haven't been able to measure it at a species level. So we've measured it at one site so far, and we found that 70% of the turtles at that site were predated, which is a really high number. You can imagine that 70% of baby turtles not actually ever getting to become adults and return to their colonies to breed and lay eggs themselves, you know, that's going to have a big impact. But we really need to be doing this at a lot more places to see how widespread it is and whether the predation rate that we measured at Thevenard Island is representative of the broader population. And this, again, similar to my response to the last question, how many students do you need to measure to know the average height of students? Well, yeah, we probably need to go and do it at a few more sites now. But yeah, it's a difficult and expensive thing to do. The tags are expensive and the logistics to do it are expensive too. So yeah, you'll have to watch this space. Hopefully we'll get to repeat our experiment, and maybe at one of those really long jetties like I showed you at the end of the presentation, where there's likely to be a lot higher impacts than at the jetty that we worked.
Okay, huge thank you, Michelle, for coming today to do the presentation. I know the kids thoroughly enjoyed it, especially the part where they saw the turtle being swallowed by a different fish and seeing that track. It's very interesting research that you're doing. So thanks so much for sharing with us. And as it's our last one of 2020, I just want to say a huge thank you to Alida Johnson at the West Australian Marine Science Institute, as well as all the other research organisations and schools that have participated. It's certainly been a fantastic opportunity, and yeah, we, on behalf of all the students that have benefited from it, huge thank you.
Well, yeah, thanks from me too. It's always a great pleasure to speak to kids, and yeah, I hope that you've got something out of it. And I wish you all the best with your studies in marine science, and hopefully you're inspired to become marine scientists yourselves.